Welcome back to Journal Club. Today's paper is looking at mysterious pancreatic islet inclusions in giant southern pouched rats. So, giant pouched rats are nocturnal, omnivorous rodents of unusual size native to sub-Saharan Africa. The pouched in their name refers to the cheek pouches, which they can use to transport large food items back to their burrows, kind of like a hamster. As the name suggests, they are also quite large, so about 70 to 80 centimeters on average, that's 28 to 31 inches, or about 4.25 bananas for the Americans in the crowd. And they are sometimes trained and used to use their incredible nose for a wide variety of tasks, including detecting tuberculosis and identifying landmines. Now, historically, their import for the pet trade was also associated with an outbreak of Mpox in 2003. This ended up getting blamed on prairie dogs, and is why you can't own prairie dogs in some states. But because of that, there's also some additional scrutiny on the populations used in research and in things like scent work. Anyway, this population was being used for research on scent work, and so they had some additional scrutiny. And so when they had these inclusion bodies pop up on routine histology, they really worked it up. So what are inclusions anyway? Basically, they are an aggregate of some sort of stable material within a cell. They can be found either in the nucleus or in the cytoplasm, and this material can be a protein, a nucleic acid, or a pigment. Sometimes inclusions can indicate things like viral infection or some sort of compromise to normal cell processes. And normally viral inclusion bodies are associated with things that make the cell sick, which are called cytopathic effect. We talked a bit about cytopathic effect in that video on papillomaviruses in polar bears. So this study is looking at 21 pouched rats, some of whom are wild caught. They are maintained in a research colony that's studying scent work. Now, some of these rats had developed diabetes, and so they wanted to investigate a potential relationship between diabetes and the pancreatic islet cell inclusion bodies. Also, with the whole Mpox thing at the back of their mind, they wanted to rule out a virus, although they had a low level of concern because it's only in the pancreatic islet cells. They have unique shapes that don't look like pox viruses, and they don't have cytopathic effect. But here are the inclusions within the islet cells of the pancreas. They are only within the islet cells, so they don't affect the exocrine pancreas, and there are both intranuclear inclusions pointed out by the arrows, as well as intracytoplasmic inclusions pointed out by the arrowheads. And so they did a variety of special stains to attempt to characterize them. So using Okajima's stain, which stains for hemoglobin, these inclusions are highlighted in bright orange. However, the periodic acid Schiff on the lower left, which would stain for glycoproteins, is negative, and the Prussian blue on the lower right, which would stain for iron, is also negative. Which is interesting with the Okajima's stain, because if it's hemoglobin, it should stain for both Prussian blue and Okajima's. They didn't show this, but they also did a Mason's trichrome, and they were light brown, and then they were not congophilic on Congo red. So taken all together, this rules out fibrin, erythrocyte material, glycogen, mucin, mucopolysaccharide, and amyloid. So we know what they aren't, but let's see if we can figure out what they are. So the next step is to do immunohistochemistry. The top two boxes are using P62, which is a marker for autophagy. Now, all the cells have some immunoreactivity because most cells are expressing some degree of P62. But the inclusions also have some speckled positivity throughout them. It's not consistent, though. They tried LC3B, which is another marker for autophagy, and it was not positive. Insulin, cytokeratin, and vimentin were all negative within the inclusions as well. Beyond me why they chucked this in the supplemental figure, because figure 1, C, D, and E are the exact same as figure 2. Same editor got on my case about number of figures, but whatever. Not salty. So the next step is to do electron microscopy, and basically all the cool kids are doing it anyway. So first step is to figure out what's normal pancreatic cells and what's abnormal. So this is a normal pancreatic cell. They lack the inclusions, but they have these roughly quarter micron secretory granules. The secretory granules have an electron-dense core, and then throughout the cell you can also see profiles of Golgi bodies, rough endoplasmic reticulum, and mitochondria. Note the normal-looking mitochondria at the arrows. The nucleus is rounded to oval with a good mix of euchromatin and heterochromatin, and the heterochromatin is mostly marginalized or around the nucleolus. Meanwhile, while we look at an affected cell, they have these inclusions composed of fibrillary, electron-dense matrix with clear spaces. They have fewer secretory granules in the cytoplasm, and looking up close at those inclusions, they are not membrane-bound. The ones with cytoplasmic inclusions have these giant mitochondria shown in B. They also have increased numbers of autophagosomes, or autophagic structures. Someone should probably go back and read Kliansky's paper on that. You can also see elongate distorted mitochondria profiles. as well as stacks of periodic crystalline structures within the giant mitochondria. So these are sick mitochondria. And then the shape of these intranuclear inclusions can also be very variable. The intranuclear ones can contain various filaments as well as vacuoles. Looking a bit closer at the one from 5D, the inclusion contains all this granular material, and then there's microtubules. So we have a great answer for the cytoplasmic inclusions. They're giant mitochondria. And these intranuclear inclusions seem to represent induction of autophagy, so they're probably related to the same process. These giant mitochondria can result from defects in autophagy, abnormal mitochondrial fission, or abnormal mitochondrial fusion. These processes can be related to oxidation or oxidative damage to the mitochondria, and we have reasons for that. Some of these animals are fed a high-fat diet during breeding, which can account for some oxidative stress. However, when they ran the statistics, the inclusions occurred in both male and female rats with and without diabetes, and even in rats that are on a low-fat diet. So unclear why exactly this is happening, and this may just represent a weird thing that happens in this species.